Now I will talk about the effects on growth and development of plants by Gibberellins on behalf of Zhang because she has some technical problems. Okay, so we have four main effects of Gibberellins on plants. That is, it stimulates stem growth in dwarf and rosette plants. It influences floral initiation and sex determination. It promotes fruit set and it promotes seed germination. The first function is that it stimulates stem growth in dwarf and rosette plants. The most pronounced physiological effect of gibberellin is the strong stimulation of prolonged growth of the body, the elongation of the internode. As you can see it in this picture, after 7 days after GA3 treatment, we have some extreme elongation here on the short stem tree. And next, the growth of a dwarf mutation. Many genes are responsible for synthesizing the enzymes in the GA synthesis pathway. You can see that after treatment of GA, it grows much taller. And this is jute. People harvest jute for something, and people spray at a rate of 20 to 50 parts per million several times for jute field, so that it can make the height of the jute double from 2 meters to 4 to 5 meters. But the jute quality is not worse. When the tree is 50 centimeters high, people start spraying 3 times. Each spray is 10 to 15 days apart. And the second function of gibberellin is the influence on floral initiation and sex determination. In many cases, gibberellin stimulates distinct flowering. The specific effect of gibberellin's flowering is to stimulate the prolonged and rapid growth of the flower cluster. Gibberellin stimulates long day flowering in short day conditions. And we have some citation here. As you can see, we have some yellow flowers here on the right, but not on the left. Gibberellin affects the sex distribution of flowers, inhibits the growth of female flowers, and promotes the development of male flowers. Gibberellin has an auxin-like effect that increases the size of the fruit and produces a seedless fruit. This effect is more pronounced when combined with auxin. And here we have some gibberellin's relationship with auxin. Auxin stimulates the larvae. GA stimulates tissue growth. Auxin stimulates cell proliferation of cells derived from metastatic tissues under the tip, prolonged. GA stimulates the proliferation of cells derived from fecal calcified tissue. Auxin against axillary buds, but the GA stimulates the shoot growth and removes some sleep from shoots and embryos. Okay, so the third function is that it promotes fruit set, increased biomass, and increase the productivity of vegetables. GA treatment increases the percentage of fruit and nuts or seeds, increases fruit size, increases fruit yield. Both auxin and GA must be present in the fruit in order to have fruit. Some crops, like grapes, cherries, have specific reactions to GA. For vegetables, increasing biomass is important. To achieve that, people often spray growth stimulators, especially GA, because GA stimulates cell growth in very strongly and is completely non-toxic because it is a natural product. For example, it is possible to spray GA for cabbage, carrots, etc., which can produce very high yields. Concentration of GA in this case ranged from 20 to 100 parts per million. For example, you can spray GA to beans to sprout evenly to increase pea yield. You soak the pea overnight in a GA 10 parts per million solution. And for grapes, one of the important directions is to increase the size of the fruits, increase harvesting productivity by using the growth promoters. GA spraying is a popular and effective way to increase the yield of grapes and to improve quality. Now the fourth function is to promote seed germination. Gibberellin stimulates germination, budding of the germ, seeds, and tubers. Gibberellin stimulates the synthesis of alpha amylase enzymes and other hydrolysis enzymes such as protease, phosphatase, and increases the activity of these enzymes, thus promoting the decomposition process. Starch into sugar, as well as decomposition of polymers into other monomers, facilitating the raw materials and energy for germination. We will see it more clearly at the end of the presentation on the signal transduction. So, to break down the sleeve of seeds, tubers, and legs, in principle, The ABA and GA balance should be changed in a way that reduces the ABA to a minimum or increases the GA. In order to prolong the inflamed period of preservation, the balance changes in the opposite direction, which is mainly the growth inhibitor. To break the sleep, people usually handle GA. GA enters the seed, which changes the hormonal balance that is favorable for germination. We soak the seeds, tubers, or spray them with GA solution at a concentration of 2 to 5 parts per million for a certain period of time. Then temper and soak can make the seed germinate. In addition to GA, many other chemical compounds are used to break down sleep, including inorganic substances such as nitric and sulfuric acids.
The next is application of gibberellin. In agricultural production, there has been multiple applications in the production and have a result to larger. The first is stimulate plant growth to increase yield as with leafy vegetable. The second is stimulate flowering fast and more, limiting flower loss, dropping young fruits, and increasing the size of fruits with fruit trees. The end is germination with dry and many other applications. For each tree group, GA can be used according to possible. For rice, GA is used to stimulate stress, to the minute, stimulate tillering, stimulate cotton at flowering, and drainage, restrict choking. For sugar cane, spraying in the beginning of the long and sluggish periods can increase productivity by 20 to 30%. GA for roots can increase tree head by 2 times. For leafy vegetables such as spinach, 2 to 3 times. The stress of strong ground can increase productivity over 30%. For coffee, cashew nuts and fruit trees, lapel, cloves, mango, rambutan, apple, milk, to stimulate flowing, rapid and smoothness. Spraying of GA when flower buds begin to form about 20 to 30 days prior to flowering or after the flowering. Drought, peeling, or spraying irrigation. GA also increase the rate of bean, fruits, and flower are limited. Other application create many flower in seed production, stimulate the salad, prolific flower in high temperature, shorten the flowering time for flower. Trees in the month. In spring, GA is used to stimulate the germination of wheat, corn, cereal, yeast production cost, increase yeast quality, and shorten yeast time. Is estimated just about 50% of the gross production of GA is used in the product of alcohol.
Bioactive gibberellins, equal GA, are not regulate absurd growth and development in plants. In an attempt to identify where GA are formed, scientists have analyzed the expression patterns of 6 GA biosynthetic genes and students with predicted roles in GA signaling and response in relation to measured level of GA. GA quantification by GC-MS-SRM so that the bioactive GA1 and GA4 were predominantly located in zones of expression of psyllium cells. Therefore, the suggests the main role of GA during wood formation is to regulate early stage of psyllium differentiation, including cell elongation. This is a figure 1 schematic overview of the Cambium region, including different types of phloem. Cambium and psyllium cells not included in the figure are the ray cells. A to Z is the approximate sample position from west section were pulled and expression analyzed. In recent years, our understanding of GA biosynthesis and signaling has increased substantially through the identification of many genes involved in this process. The biosynthesis of GA can be divided three stages. The first one is the formation of N current. The second is the conversion of N current to GA12, and the last one is the formation and deactivation of bioactive GA. The enzyme that undertakes GA formation is a proplastid located and covalent diphosphate synthase. This is called CPS, and which catalyze the conversion of geranyl diphosphate GGPP to covalent diphosphate CDP. Covalent diphosphate is subsequently converted to N current by action of N current synthase, is called KS. To membrane-associated cytochrome P450 monoxygenase that then catalyze sequential oxidation of N current.
Zeaxanthin mediates blue light photoreception in guard cells. The carotenoid zeaxanthin has been implicated as a photoreceptor in blue light-stimulated stomatal opening. Zeaxanthin is one of the three components of the xanthophyll cycle of chloroplasts, which protects photosynthetic pigments from excess excitation energy in guard cells. However, the changes in zeaxanthin content as a function of incident radiation are distinctly different from the changes in mesophyll cell.
And the second
Of guard cell
And select
carbohydrate
one.
metabolism with membrane ion transport during stomatal opening. Sugars in guard cells can be imported from the apoplast, derived from starch breakdown, or be synthesized in the Calvin cycle. These sugars then can be stored as osmotically active solutes in the vacuole or metabolized in the cytosol to yield energy, reducing equivalents. And phosphoenol pyruvate, PEP. PEP can be further metabolized. Pyruvate and the mitochondrial tricarboxylic acid CAC cycle are used as carbon skeletons for the biosynthesis of malate via PEP carboxylase PEPC and NAD-dependent malate dehydrogenase NAD-MDH. Malate, which also can be imported from the apoplast, and the inorganic ions K plus and Cl minus accumulate in the vacuole, lowering the guard cell osmotic potential, thereby promoting stomatal opening. ABCB14. ATP binding cassette transporter B14, acetyl-CoA ALMT, aluminum activated malate transporter, ATP-VFK, ATP-dependent phosphofructokinase, ATTDT, dicarboxylate transporter, CINV. Cytosolic invertase, CWINV, cell wall invertase, FRU6P, FRU6P, FRU1, 6P2, fructose 1, 6-bisphosphate, GL6P, GLC6P, G3P, glyceraldehyde 3-phosphate, IPCM, phosphoglycerate mutase isoforms, NRGA1. Negative regulator of guard cell after signaling 1, OAA, oxaloacetate, 2-PGA, 2-phosphoglycerate, 3-PGA, 3-phosphoglycerate, PPI-PFK, PPI-dependent FRU6P phosphotransferase, STP. Monosaccharide, H plus cotransporter, SUC, H plus cotransporter, SUSI, SUC synthase TPT, triose phosphate, phosphate translocator. Compartments are not to scale. The dotted line indicates multiple metabolic steps.
and the secondary the bupal
While the removal of Cl- and K+ is well described in the literature, the fate of SUC and malate during stomatal closure is unclear. SUC can be cleaved by cytosolic invertase, CINV, and the resulting hexoses can be imported into the chloroplast in the form of GLC6P. GLC6P is used subsequently for starch biosynthesis. Malate can be removed from the cell via decarboxylation to pyruvate by malic enzyme, ME, and the subsequent complete oxidation in the mitochondrial tricarboxylic acid CAC cycle. Alternatively, malate can be converted to PEP-via NAD plus dependent malate dehydrogenase, NAD-MDH, and PEP carboxykinase, PEPCK. Gluconeogenic conversion of PEP to GLC6P establishes a possible link between malate removal and starch synthesis. Compartments are not to scale. PEP, phosphoenol pyruvate, OAA, oxaloacetate, STP. Monosaccharide, H plus cotransporter, SUC, H plus cotransporter, SUSI, SUC synthase, CINB, cytosolic invertase, NRGA1, negative regulator of guard cell ABA signaling 1, ALMT. Aluminum activated malate transporter, GPT GLC6P, pi translocator, CWINB, cell wall invertase, HK hexokinase QUAC1, quickly activating anion channel 1.
Let me talk about the physiological mechanism of Gibberellin-induced growth. There are three main ideas that we are going to follow. The first is Gibberellins stimulate cell elongation and cell division. The second one is that Gibberellin enhance cell wall extensibility without acidification. And the last one is that Gibberellin regulates the transcription of cell cytokinase in endocrine meristem. We begin our discussion with the role of Gibberellin in regulating cell elongation. Could you know some characteristics of Gibberellin-induced stem elongation? Gibberellin increase both cell elongation and cell division, as evidenced by increase in cell length and cell number in response to application of Gibberellin. For example, internal of tall bees have more cells than those of broad bees, and the cells are longer. Mitosis increase markedly in the cervical region of the meristem of rosette longleaf plant after treatment with Gibberellin. As we know, the elongation rate can be influenced by both cell wall extensibility and the elongation rate can be influenced by both cell wall extensibility and the osmotically driven rate of water uptake. Gibberellin has no effect on the osmotic parameters but causes an increase in both the mechanical extensibility of cell walls and the strict relaxation of the walls of living cells. An analysis of peer genotypes differing in Gibberellin content or sensitivity so that Gibberellin decreased the minimum force that will cause wall extension. Thus, both Gibberellin and auxin seem to exert their effects by modifying cell wall properties. In the case of auxin, cell wall loosening appears to be mediated in part by cell wall acidification. However, it is not the mechanism of Gibberellin action. In no case has a Gibberellin-stimulated increase in proton extraction been demonstrated. On the other hand, Gibberellin is never present in tissues in the complete absence of auxin, and the effect of Gibberellin on growth may depend on auxin-induced wall acidification.
Oh, hello people. Now let's talk about the signal transduction for Gibberellin. Gibberellin, featuring the aileron layers. Now let's talk about this aileron layer. Now we have this thing is the seedling, and the seedling we have the embryo. The embryo is the thing that's going to develop into a tree, and this one is the endosperm. It contains the nutrition like starch and other macromolecules that are broken down into nutrition for the embryo for growth and development. And this is the testa pericarp. It is just the covering of the embryo. I mean the endosperm. Now this is the aileron cells on the aileron layer. They do the work. Their function is to produce hydrolytic enzymes into the endosperm that convert from starch, for example, to glucose, for example, for the use of the embryo. And we know this because we know that the embryo secretes GA into the endosperm Gibberellin because when we remove the embryo, degradation of starch and molecules does not occur. And we know that the embryo secretes something diffusible into the endosperm, because when we put the embryo in close proximity with the endosperm, but not touching it, they do not touch each other, then degradation doesn't occur. And finally, we know that that substance, that compound, is gibberellin, because when we introduce the endosperm into a solution of gibberellin, then degradation does occur. And so, in conclusion, in general, the embryo regulates its own nutrition intake by secreting GA, gibberellin, into the endosperm, and the aileron cells will receive those signals and produce hydrolytic enzymes, typically alpha amylase, into the endosperm, and to convert from starch to glucose, for example, that is going to feed the embryo. Now let's talk about the signaling pathway of the aileron cells here. This is the chart, the illustration from the textbook, but it's a little bit small, so I'll enlarge it like this. And let's go into it now. We know that the GA receptor is on the plasma membrane, because when we attach the GA to a microbead that is unable to cross the membrane, the production of alpha amylase still occurs, so it means that the receptor is on the outside. Another evidence is that this is the protoplast. It contains proteins for the production of, as the material for the production of alpha amylase. And when we insert GA3 into it, nothing happens. So so the signaling does not occur inside these protoplasts. But when we immerse them, into a GA3, then production does occur. It means that the signaling is outside these protoplasts. And next, this is the G protein, the cyclic GMP, and we know that it contributes somehow to the pathway, because when we introduce GI, then this is the diagram for the response to GI over time. And we see that the cyclic GMP is stimulated by GA here. And here, look at this. This is calcium. The calcium plays some role in the secretion of alpha amylase, but not in the production. We know that because when there is no calcium, then production does occur just as normal, but the secretion does not occur. Okay, so that's for calcium. Just secretion, not production. And next, we know that there is some intermediate protein in the pathway here, because when we introduce some protein that competes with this protein in terms of activation of activity, then production does not occur. Actually, production is inhibited. So that is the evidence. And the activation of this protein is probably by phosphorylation, just like when we add the phosphate group to ADP to become ATP. And now about the protein production here, we have these two genes. Genes. Just look at this graph. You see that the alpha amylase transcription is like the curve is like a shift from the GA, MIB, mRNA transcription. So there are like two linear steps on the process. And we know that GA1, gibberellin, stimulates the production of alpha amylase, not just the secretion, because when we introduce heavy isotopes or radioactive isotopes like carbon-14 into the materials for production, the secreted alpha amylase does contain those isotopes. It means that they are synthesized, not just that they are already there and then secreted. And now let's talk about the production of alpha amylase. Now you see that this intermediate protein will bind to the repressor to release it, and transcription can occur. This is the RNA polymerase, and it will transcribe the gene into mRNA, and the mRNA will be translated by the ribosome tRNA into the transcription factor. Here is a protein, and this protein will again stimulate the transcription and translation of the alpha amylase gene like this. Polymerase and ribosome, and the ribosome here is attached to the rough ER outside the nucleus. We know that the GAMYB gene contributes to the process, because when we introduce the factor that inhibits translation, we see that GAMYB transcription occurs, but alpha amylase transcription not translation, does not occur. That's because there's no translation, so there's no transcription factor for the alpha amylase. Okay, that's the end. Thank you for listening.